This is a favorite of mine, so I'm glad to share this topic with you because I find it exciting. I mean, sex. What's there to not like about sex? You know, it's just, you start out with sex and you know it's gonna get better from there. So if you were at my lecture this morning, I talked about one of those pivotal moments in the evolution of bees is when the females started practicing polyandry. And if you remember this diagram, you can see it up a little closer now maybe. The point that I was making is that it's a general rule of evolution that there is a lumping of lower levels of order into higher ones. You know, loose nucleotides get lumped together into chromosomes, lumped together into cells, into multicellular organisms and superorganisms. And the jump from the multicellular organism like you and me to the superorganism requires polyandry. It's a necessary step in that process because what you get is this division between the reproductive group and the rest of the group, the queen versus the workers. In our case, at the level of an organism, we're talking about like our testes and ovaries, the reproductive part versus the rest of our body. That's the distinction we're talking about. And in the superorganism, it's the queen and the drones who are the reproductive organs, and the workers are the somatic cells in the body. So that's kind of the concept that I've been talking about here. So if we're going to try to do breeding with an animal at this level, it's going to look different than breeding at an animal at this level. And we are all accustomed to breeding at this level, right here. You know, we breed cows, we breed horses, dogs, corn plants. You know, they're all organisms. And one thing that's unique about the multicellular organisms is their cells are clones of one another. Your cells in your liver have the exact same set of genes as the cells in your lungs. They're all clones of one another. And this is one of the things that this organism has used to keep down mutiny, that rebellion of these lower levels. They've just gone through this bottleneck where they're all clones. And so that's why most of the time your liver is not in conflict with your lungs, or vice versa. You know, all of our cells are in harmony with one another because they're all clones of one another. The superorganism is not clones. The worker bee is not a clone of her mother. So things are a little more chaotic and disorganized at this level. It's harder to breed at this level because of that reason. Okay, so that's very important to understand in how we breed the superorganism. But whether it's superorganism or organism, the vehicle of all of this, of course, is the chromosomes. And what I've shown here is a chromosome. A chromosome is a really tightly wound string of DNA. And we know this famous molecule, the DNA, where all of our genes are housed. And it's like a ladder, and then you twist that ladder. And that's the classical structure of the DNA molecule. But a chromosome is just simply a big, long section of DNA that's closely wound up to one another. And they typically occur in pairs in a diploid animal like you and me. And a diploid means we have one set from our mother, one set from our father. This is what we're used to. And for diploid animals, that number of chromosomes is unique to the particular animal. In the honeybee, it has 16 pairs for a total of 32. You get one pair from your father, one from your mother. Typically, I'm just kind of showing it here, this set is from the father, this is from the mother. The chromosomes tend to line up in pairs with one another. They're roughly the same size as one another. We call this a homologue of this chromosome. One comes from the father, one comes from the mother. And they carry different forms of particular genes on these chromosomes. You get one form of it from your mother, one from your father. Humans, we have 46 chromosomes. We have 2 pair of 23, so it all adds up. But there's an exception in the Hymenoptera where there's only one side of the chromosomes, and that's with the males, right? Males are across the Hymenoptera, they have only one set of chromosomes. And this is true. Now let's talk here a minute about how inheritance happens in the and this is going to be kind of a review of your biology from high school or college. Gregor Mendel, the great monk who figured out the mathematics of inheritance. The guy was absolutely brilliant. I mean, just imagine this. He did not have molecular knowledge nor capability. He just figured it all out just from intuition and applying mathematics. And Gregor Mendel, he was this monk in the modern day Czech Republic, he liked pea plants, and he studied, forgive me here, I'm forgetting, wrinkled peas versus smooth peas, and yellow flowers versus blue flowers, was it? There were certain characteristics in peas that he did all of these crosses, and he figured out with extreme precision which cross would make what. And he's the one who figured out the laws of inheritance before we even knew there was DNA and chromosomes. Now this is another one of those historic what-ifs. Gregor Mendel and Charles Darwin were contemporaries. They were alive at the same time, Mendel in Czech, Darwin up in England, but there's no record that they ever knew each other. There's no evidence that they ever met or was aware of one another's work. And it's one of those what-ifs. You know, if Darwin and Mendel had met, they probably would have pushed biology forward by decades. But we know from Mendel that, you know, the gene occurs in different forms, one from your mother, one from your father, and I've just marked a section of this chromosome just to roughly designate a gene. And let's call that gene the gene for determining sex. Now, if you've read all the books will tell you that bees that have one pair of chromosomes are males. Bees that have two sets of chromosomes are females. And functionally, that's true. I mean, nobody's going to argue with that. But we know that's actually a little more sophisticated than that. You can inherit your sex just like you inherit any other character if you're a honeybee. And the sex gene actually has, I think, currently about 19 different forms. We call those alleles of the sex-determining locus. And that's a lot of alleles. And anytime we have the different forms of that allele showing up on one chromosome and showing up on the other, we call that situation heterozygous. Hetero means different, heterozygous. If you have the sex gene with only one pair represented, we call it hemizygous, and that means half. Hemi, they only have one. That too will be a male. But what if it's homozygous? What if you just by mathematical chance happen to get both forms of the sex locus in your mating with sperm and egg coming together, and you have a homozygous situation? And what do you have in this situation? You also have a male. A male. But you never see them because the bees, the workers, eat them. These homozygous males get eaten up. So they never appear in the population. Imagine there'd be some very bad things happen if these diploid males were turned loose into the breeding population. You'd probably get these lethal matings that wouldn't work. But what do you see as a beekeeper? You see spotty brood. And this is an important point. You know, whether you breed anything with honeybees, when you see spotty brood, it could be disease, it could be some environmental thing, but it could also very likely be a sign of inbreeding depression. And that's what inbreeding is. More and more of the same alleles showing up and down the genome. And if you're getting homozygous at the sex locus, you're probably getting homozygous at a lot of other loci as well. And it's one of those quasi-rules of biology that nature doesn't really like a lot of homozygosity. These individuals, we call them inbred. And that's what it means to be inbred. My mentor, Mr. Paul Champ, he really was a champ. He was a great guy. And you all got mentors in your memory, and they're all champs, aren't they? Well, Paul Champ was mine, and I remember vividly going around with him. And we go to one of his little yards and say, "Oh, yep, got a queenless hive here, Keith. See, bad brood, they're worker eggs. And okay, what we're going to do right now, Keith, we're going to go over here and get a frame of brood from this hive here, open eggs and larvae. Oh, yeah, that's good. We're going to stick it right down in here, and they're going to raise their own queen." I thought, really? He said, "Yep, that's all you got to do?" "Yep." Cool. Well, fast forward 10 years, and I go off to graduate school at LSU. And then was the first time in my life I realized how crappy Mr. Champ's bees were. They were sorry. Spotty brood all over the place. I never saw a good frame of brood till I went to LSU, because Mr. Champ, that was his only way of requeening. You know, every single time he found a queenless colony, he'd just go over to the nearest neighbor and give it a frame of brood. And after 40 years, you know, they were all inbred. Yeah, horrible inbreeding. So this is practical. I mean, this is very, very real. So it's important to keep in mind that when we as humans practice artificial selection, there's nothing really artificial about it. I mean, selection is selection. What makes our selection different from natural selection is we don't have the abundance of time to correct errors. And we're also very myopic. We've got one thing in mind. I want Varroa resistance. And I'm going to breed for Varroa, you know, and to hell with anything else. Oh, honey, brood, you know, I'm just Varroa. That's what I'm after. Well, when we apply our artificial criteria on one or few things, selection is a zero-sum game. You know, there's only a certain number of alleles out there, and if you are actively selecting for this, and this, then you are by attrition losing other things that are as important, if not more important. Natural selection has the luxury of time to even out those mistakes. Oh gosh, look, it's a dearth year. And oh, all of the bees who can't conserve their food, they die. And oh, next year it's a great year. And this goes on and on and on until the species is shaped, literally the extremes are winnowed away, so that the species is shaped to express an optimum range of genes that cover most contingencies that that habitat throws at it. That's the only difference between artificial and natural selection. Natural selection can correct for errors and excesses. In any given generation, that excess can be corrected in later generations. That's the only difference between the two. But natural selection is, of course, better, because it has more time to winnow out those errors and arrive at optimum combinations for a particular habitat. But inbreeding is the bête noire of honeybee breeding, because when you're selecting for Varroa, you are by attrition losing, or ignorant of, blind to other characters that you could be losing that are at least equally important. So I'm going to be talking about some ways that we can, you know, manage this. What you're seeing here is a bullet that has a lot of information. Let me cover it. These are different ways historically that we can breed honeybees. And the first bullet is what we almost all do. When we say we're breeding for honeybees, we're probably talking about the first bullet. And this is maternal selection. Why maternal? Because I'm looking at the colony. The colony is her direct expression, and I'm going to propagate that queen. And this colony has what I like. The bees are pretty. They're gentle. They're making lots of honey. They're, you know, all those kind of things. I'm going to breed from this queen because I like her. So you go in there and you graft. Well, now you know, because of polyandry, that the character that you're interested in may be represented by 1 out of the 20 drones that she mated with. It's just, I'm pulling numbers out of thin air here. So that means, if that were the case, then mean only 1 out of every 20th egg that you're grafting has the character that you're interested in. So is this slop or is this slop? Okay, well, let's do this. Let's graft them. And now what are we going to do? We're going to raise these cells. We're going to put them in mating nukes. And now what? Well, go mate. You know, mate with the good ones. You know, good luck. And off they go. And we wonder why breeding is so slow. You know, there is no other animal breeding system that would tolerate a mess like this. But this is what we do, isn't it? This is what 90% of bee breeding is. Now, lest you despair, let me point out 2 other things. Not all characters are equally heritable. And by heritable, we mean responsive to artificial selection. And this is one of those great biological what-ifs. It just so happens that in Apis mellifera, gentleness and honey production are highly heritable. So you can exercise a sloppy mode of selection like I just described and succeed. You can get away with it. totally by virtue of the high heritability of the two characters that matter to us most. Now, is that serendipity or what? I mean, I honestly think that if that were not true, there would probably not be beekeeping today, because not all characters are equally heritable. I can give you an example. Domesticity appears to be one of those serendipitous characters that occurs in some species and not in others. Zebras. I am no equestrian, but people promise me that you cannot tame a zebra. You can't ride them, you can't bridle them, you can't domesticate a zebra. Why not? It's just a horse, isn't it? A horse with a bar-coded horse. But no, apparently not. It does not have domesticity in its tool bag, and it does not respond to human management. Honeybees do when it comes to honey production and gentleness. And so that has enabled us to have this ongoing relationship with them that is now managerial more than predatory like it was in its early evolution. Remember me talking in my opening lecture this morning how, as a demonstration of the plasticity of defensive behavior, we both selected for their defense behavior and we have selected against it in historic times. So their toolkit of defensive behaviors, the barbed sting and the ferocity of their attack, is because of us. And their gentleness is because of us. And that selection is more recent, probably only about 7,000 years old. I just find that really cool. So that's bullet number one. Bullet number two takes advantage of that inbred hybrid situation that I talked about early, and this is what we see in a lot of plant breeding. Corn, hybrid corn is all about this. And it's that idea about nature likes heterozygosity. In general, an organism, you or me, if we have a lot of different forms of the genes, different forms of that allele that we inherited from our mother and from our father, we'll tend to be a vigorous, healthy individual. This is usually true. There are exceptions. And one of the most interesting exceptions for us is African bee and European hybrids. This is one of those examples of negative heterosis, where the hybrid is less vigorous than the parental type. And hybrid African European bees just don't persist. They mate, they breed, they exist, and ecologically they just fall off the cliff and they disappear. And this has been shown over and over again in their history coming up from Brazil since the 1950s. It's been one of the engines that drives Africanization, the negative heterosis of hybrids. But that's the exception. It's funny how honeybees always have so many exceptions that form the basis for a principle that's then applied across all of biology. That's the kind of animal that we're working with. But in the inbred hybrid scheme, Dadant actually marshaled this principle back in the 60s and 70s. Some of you may remember the midnights and the star lines. And what Dadant did was maintain a breeding station down in Florida, and they created these highly inbred parental lines. And how do you create an inbred honeybee parental line? Well, one thing you can do, you can gas a virgin queen with CO2, and CO2 with virgins, like it does across all insects, it generates egg-laying behavior. Now, she's a virgin, she's not mated, so she produces males, so you see where I'm going with this. You rear those males to maturity and then turn around and inseminate her with her own sons, which is essentially her own gametic cells. She's inseminating herself with herself. Selfing. Now, is that weird or what? So what you get then is a huge quantity of homozygosity across her entire genome. On both sides of the genome, lots and lots of mathematical sameness going on. And so you get these sorry bee lines, kind of like Mr. Champ. They're just really, really bad bees. But when you cross them, boom, what do you get? You get maximum heterozygosity on both sides of the aisle. And you get these hugely productive, vigorous bees. And that's what the midnights and the star lines were. And I'm old enough to remember, I worked them. I got them when I was a kid. And they were nice. They were nice bees. They were gentle, made great honey crops. I mean, they were a great bee. But then along comes Varroa mite in 1987. And midnights and star lines just did not have the tools to resist this very unique, singular threat that they have never encountered in their phylogenetic history. Remember my talk this morning about the latest data demonstrate or arguing for a European origin of the genus? And then we had that lineage that immigrated out of Europe down into Southeast Asia. I'm trying to do this. Okay, Southeast Asia, okay? And then we had that group that came down over Spain, down into Africa, the Western lineage, which gave rise to the mellifera that we have today. Mellifera never encountered an Asiatic mite, never, in its phylogenetic history. So the inbred hybrid scheme was to no avail. It needed something more precise, something more narrow, something more deliberate, and this is why selecting for resistance has been so hard with Varroa. It has no tools that are native to Asiatic mite resistance. And any tools that it has are co-opted from other behaviors that we hope we can now turn their attention toward mites. And I talked about this morning grooming behavior, which is probably ancestral to grooming off the bee louse. And we're hoping and trying to get it oriented toward the Varroa mite now with our breeding efforts. Hygienic behavior oriented toward bacteria and fungi, brood diseases, and we're trying to reorient it toward Varroa. So you see the difference? We're taking an alien trait and trying to twist it to a new and novel problem, and the success, well, speaks for itself, doesn't it? Speaks for itself. It's been really hard to find resistance to Varroa in Apis mellifera. Okay, let's go on to the third bullet. This is the most sophisticated method that's out there. This was developed by Rob Page and Harry Laidlaw back in the 1980s, closed population. This is worth knowing. Okay, if you're a breeder, you might want to listen up because this is practical and kind of the state of the art. And it's all in the name, closed population breeding. So you got a population that is closed. You do not want introgression of new stock. And you want to have at least 50 colonies. You got to have a certain viable population to avoid inbreeding as long as possible. And the recipe is pretty simple. Once you've got your defined population of 50 queens and colonies, then you select for whatever it is you want. And you select the top 10% of your colonies and you graft from those top 10%. Okay, so far so good. But now you inseminate those virgins with a pool of semen representing the top 10% plus all of the bottom 90% too. So it's like taking 2 steps forward and then 1 step back. You're kind of losing some of your selection. Then the next generation you do it again. You get another 2 steps forward and another step back. And they admit this is a compromise. This is an attempt to select towards something that you want while at the same time minimizing inbreeding. And they actually, it's a very sophisticated model. They predicted that you can get about 20 generations of this before you start seeing inbreeding at a scale of about 85%. In other words, you're starting to lose 85% of your brood because of inbreeding. But you know, for most of us, 20 years is a realistic horizon. I'm probably not going to be breeding 20 years. Well, maybe I will. Don't short credit myself that way, but you know, 20 years, it's not a bad horizon, is it? So this is the state of the art, and this is what Sue Kobe uses with her New World Carniolans. I think some of the guys in California are using this in their scheme. It does require either instrumental insemination or islands in isolation, but do not despair. I had one of my master beekeepers last month point out to me that you can now download printable AI devices. You know, we always worry about, oh, they're too expensive. Well, not anymore, they're not. You can now 3D print your own insemination device. So this is one of my soapboxes. I really encourage clubs to invest in an insemination device. You can learn it. It's like learning a bicycle. You know, you got to learn, but once you learn, then you know it. So I do encourage clubs to consider this because it's a great tool and opens up vast resources to you. And you can now print them for practically nothing. So there you go. That's the 3 kind of standards. Now, whatever of these methods you're using, well, except for the inbred hybrid. The inbred hybrid is not a selection, by the way. That's just sort of let's just make them hybrid. And when they're hybrid, they're going to be good bees. Okay, there's really no selection. But for For maternal, the first bullet, and closed population, the third bullet, you do want to engage in some selection. If you're interested in weight gain, you want to weigh colonies. If it's brood area, you want to measure brood area. If it's defensiveness, you want to count stings. You want to empiricize it. You want to actually count, weigh, and measure to reduce, your slop. The system's already sloppy enough. Heaven help us. Let's not, interject anymore. And of course, with mites, you would, count those colonies with the fewest mites. Okay, well, this is great. So now the question is, that's how we breed the honeybee. And let's ask now, how does the honeybee breed itself? Or how does the honeybee marshal genetics? That's the point of this talk. And I have already mentioned how the superorganism operates by different rules. We have to apply some different criteria to our breeding programs when we're talking about a superorganism. If there's one thing that's absolutely true about the honeybee, it is outrageously committed to genetic variation. Radically so. I can even attach a superlative to it. What you're seeing here is a step in the formation of gametes, which is elegantly called crossing over. And this is this is happening in the testes or ovaries of males or females. And remember, we all start out as diploid animals. You and me, we're diploids. The bees are weird, but in the honeybee queen, this is what's happening. In the male, this happens without crossing over because there's nothing to cross over with. Remember? So the male is kind of an oddball, but for a diploid female, the two halves of the chromosomes line up, the two homologs that are similar size to one another, they cross their legs. Saxophone music's playing again here. The sections of the chromosome break off and they reconnect and then pull back apart and then go ahead and form their separate gametes. And this is a form of exchanging chromosomal material in the very formation of the egg or the sperm. Well, this is one level. And this, of course, we got to think about him. How does he contribute to variation? The drone congregation area. The drone congregation. This is where the male participates in this process. And they make these flying comets. There's no other word for it. You remember Bugs Bunny and the Wile E. Coyote? I forget which episode, but there's an episode where these bees are chasing Wile the coyote. And that's exactly the way they act. They really do act like that. There's this comet that just chases the queens. And this drone congregation area can be quite large over a scale of square miles. And it's literally the guys just cruising the strip. And they got one thing on their mind. And how do they form? Well, they're another one of those emergent properties that I talked about this morning. People have wondered what makes a DCA, drone congregation area, what makes them so permanent? I mean, they last year after year after year, and it's an example of intergenerational learning, right? Some of the drones survive winter and they pass on the secret handshake to the next generation. And no. It's simply driven by landscape features. Linear landscape features. And if you are a drone and you fly out on a beautiful morning and you survey the landscape, there's going to be something to orient to. It may be strong linearity. It may be a margin of a woods. It may be a horizon window, a dip in the horizon that has light coming out of. There's going to be something out there for you. And they're going to go out there, and there's going to be, within drones in a similar area, are going to have a greater likelihood of encountering those same landmarks that kind of channel them together. And then there tends to be the power of suggestion. Have you ever come to a bee meeting and you didn't really know where the meeting was going to be? And you show up and you park and you get out of your car and you just see these guys walking. And you know a beekeeper when you see it, right? There's just something about them. Yeah, that's a beekeeper. And what do? You kind of follow them. Well, that happens with bees and animals too. They see other guys cruising the strip, so they kind of, hey, what's going on? I don't know. Did you hear? I don't know. I'm just hanging out. So they fly around, and that's how they start. There's nothing mystical. It's the exact same process that literally makes a small town American high school gaggle of guys cruise up and down the strip. I'm dead serious. It's exactly the same geography that makes a DCA happen, except it's the main street. In Logansport, Indiana, it was Market Street. It was Market Street and Linden, and they would just cruise the strip. Of course, I wasn't doing that, but they'd cruise the strip. It's just geography. There's been some elegant work in Germany that's tried to figure out these DCAs, and their data are staggering. An average DCA has drones coming from at least a 7-kilometer, like about 5-mile radius, and over 240 colonies. 240 colonies contributing to one DCA. Just staggering. Variation, variation, variation. And it's adaptive for both the males and the queens. For the queens, flying is risky. A bird might pick me off. So I am going to try to fly as few times as possible, but I'm going to try to pack in as many copulations as possible in each flight. Each flight is risky, each copulation is not. Now, I'm already airborne, I'm already in this DCA. The more the merrier. Come on, guys. Boom, boom, boom. But to go back home and go out on another flight is mathematically more risky. Flights are the danger, but once you're airborne, try to get in as many as you can. And DCAs help them do that. Help them do that. And if you examine, we did DCAs when I was a graduate student at LSU. We were interested in African bee mating behavior and how we could make more of the right kind of matings instead of the bad kind of mating. So we were working with DCAs. And they literally formed that comet. And we had this big long line of graduate students down this power right-of-way, and every mile there was one of us, and we had these goofy little traps. They were like 20-foot high aluminum poles with what looked like a trash can suspended from the top, open at the bottom, which was made out of really light gauze. I mean, the whole thing was just feather light, really, really super. And dangling from the top of that was a little black piece of chalk soaked in queen pheromone. And these drones would come whoosh, and you could literally hear in the sky over your head, and they'd land on your head. Dead. Dramatic. But queens can really rapidly get many copulations in this kind of a situation. The drones do not fight with one another. To the contrary, they appear to cooperate with one another. You've probably heard about the mating sign, where the endophallus of the male literally breaks off. And this is a primitive state of blocking subsequent matings. It's all about me. You can't mate. Well, in the honeybee, the guys are much more civil and sophisticated than that. They say, no problem. In fact, yeah, this is better. I mean, I want more males to follow after me. Because if my mate has lots of males represented in her spermatheca, that superorganism is going to be genetically diverse. So this diversity is still the end goal, even though it looks different in a superorganism. And male honeybees have adapted these little hooks. They slide in under the endophallus of their predecessor. There's these little hooks, and they kind of flip it out and jettison the last guy's endophallus and stay in there, breaks off, next one, breaks off, next one, so that when they get back to the nest, when the queen gets back to the nest, the workers have to take the last one out. But these mating signs stimulate and help subsequent males find that same queen. Males that have a mating sign, queens that have a mating sign are more attractive to drones than queens without a mating sign. It's counterintuitive, isn't it? But the males are saying, I want her to have a lot of other guys because this will increase the fitness of her colony. Is this cool or what? Right, yeah. Oh, I got to tell you this. This is for free. This has nothing to do with anything. But I got a friend, a buddy in Wales. His name is Carl Scholler, and he's very active. If you know about British beekeeping, you know about Carl Scholler. He's written a lot of books on the subject, but he's made a lifelong habit of following DCAs. And this is the way he does it. He has a big, long cane fishing pole, and he dangles a little lure from the end of it. And he just goes walking around the neighborhood. By now, the neighbors know about it. But he's discovered and mapped all these DCAs for his long life. Well, he has a theory of the origin of leprechauns. That is the best theory of the origin of leprechauns I've ever heard. And he says, imagine that you are a Neolithic or ancient Brit, and you are at one of these standing stones that litter the landscape of Britain, like Stonehenge. And there you are, you're having your mystical moment. And suddenly, out of the corner of your eye, whoosh, whoosh, whoosh. What was that? Well, it was a leprechaun, of course. You were visited by one of the little people, always out of the sight, always on the edge of your vision. But we know, of course, probably a DCA because they're attracted to landmarks like standing stones. Hey, I'm sticking with it. I'm in. That's where leprechauns come from. So there you have it. Yeah, and there we are, just to prove the point. That's Dewey Caron. I think some of you know Dewey, and then Michael Young, who some of you may know from his work over here in the States promoting honey judging. And there we are at a dolmen right outside of Belfast, looking for leprechauns. Right. All of this is accomplished by the queen's habit of multiple mating. Inside the queen, in this imperfect photograph, we have nestled this organ right here called the spermatheca. And it is an adaptation of the median oviduct, been invaginated and complexed to house semen. The semen are actually injected into the lateral oviducts, which are these organs right here. This is the median oviduct, and the drone is explosive. You know, if you've popped him in your hands, you know what I'm talking about. It's literally explosive. One of the reasons why he dies, because he's injecting the semen deep into the median oviduct, into the lateral oviducts, and these things are very elastic. They can stretch like balloons, and the queen has stretch receptors inside these lateral oviducts, and when it feels filled up enough, whatever enough is, then she, intuits that she's had enough matings, and that's when she will terminate and come back to her nest. Over for the next 24 hours or so, the sperm migrates. 97% of it goes right out and is lost. But if she's mated with enough males, there'll be a significant number that get into the spermatheca, on average about 5 million. And that 5 million is enough to satisfy her an entire lifetime's worth of egg production. Curiously, the semen of one male produces 5 million sperm, but she wastes 97% of it. So it's like, you see what's happened? She has been adapted to be inefficient. The ancient inefficiency of Hymenoptera has not been done away with. They've just tweaked the system with polyandry. And if she's wasting 97% of the sperm of 20 drones, she's still going to end up with a viable number in her spermatheca, number-wise, that is now genetically diverse. Isn't that amazing? And she comes up with a viable quantity for her lifetime production, which is probably about 600,000 eggs, and that 5 million will amply cover it and be genetically diverse. I think you're seeing a theme here, diversity. Oh, I got to say this about the Brits. I was speaking about this last fall. I was at the Devon Beekeepers Association. This is right outside of Buckfast Abbey. They own Buckfast Abbey. That's their thing. So if you ever go there, you will meet the Devon beekeepers. And I was talking about this, and I said, yeah, and the spermathecas, I don't know how they managed to not get tangled up, because the sperm of honeybees is the longest in the animal kingdom. It's like itsy bitsy head, really long tail. And I just don't, this is me talking, I just don't know how they keep from getting untangled. And the beekeepers, oh yeah, we've studied that. And I say, what do you mean? You say, oh, well, some of us got interested in what the spermatheca looks like, and so we got one of our members who's a veterinarian, and she volunteered her lab equipment, and so we've been meeting for the last 6 months, dissecting spermathecas and sectioning them and photographing them. And I'm just sitting here, that would never happen in Georgia, maybe Texas, but. So anyway, this photograph is courtesy of the Devon Beekeepers Association of a spermatheca. It has one layer of cells around the outside, so it's a single cells, and all of this is the tangled up sperm in the middle. And somehow they do not get tangled up. The use of sperm is rather reckless. It's up to about 30 sperm are let go for every egg. So her wastage is pretty rapid. But yet still, with 5 million, she's got enough for that critical limit of about 600,000 drones that she needs. Okay, in the interest of time, I'm sorry, I got to keep moving. Let's talk a little bit about how much polyandry. Why do they do polyandry? What is the benefit of polyandry? We always say, well, genetic diversity. Well, yeah, What does that mean? What does it mean to be genetically diverse, and why is that important? Well, if you look at genetic diversity, it's pretty easy to mathematize that. If you look at the breeding population of a particular DCA, there is a finite number of alleles. Okay, they're not infinite. The species is infinite, and any population of the species is going to be even less infinite. So let's just say we've got that number. And if you look at the probability of capturing all of the alleles based on the number of matings, the line flatlines at about the sixth mating. It's weird. After 6 matings, she has captured 90% of the alleles in her breeding population. After only 6 matings. But she habitually doubles that and goes beyond. Why not just stop? Okay, 6 males, boom, boom, boom. Birds, danger, danger, let's stop. No, she doesn't stop, she keeps going. We have records up to 12, up to 28, up to 40, and just last summer, to my knowledge, the highest number was published, 77. Why? Why should she do that if she's already captured all 90% of her alleles in the first 6 matings? Well, the best thought right now is that there's this possibility that rare beneficial alleles, well, let me say this differently, alleles that are really bizarrely beneficial will be rare, which is counterintuitive. Have you ever wondered in your personal beekeeping, when you buy a hygienic or a groomer queen, why does the trait go away? Why do you have to keep buying that? Have you ever wondered about that? If it's so good for them, why didn't it stick around?
Does it stick?
Well, it may not stick. Yeah, that's part. But the reason I'm talking about this rare thing, there's a team in Germany that said, wait a minute, we predict this to be the case. And they gave the mathematics and the genetics behind it. We predict, and here's how it works. If you have a really potent, serendipitous allele that makes you wake up in the morning and you're 30 years old again and virile and, wish that there were, right? Well, there's this serendipitous trait like having Varroa resistance if you're an Apis mellifera will be rare because it is so potent in its selection power that it will, just by the sheer overwhelming dominance of this one character, be ignorant to other alleles that are more important, like oxygen respiration, sugar metabolism, other fundamental things. It's counterintuitive, but it makes sense. It could be so good for this one thing that it would just blank out over other things that are more important. There's 2 examples I think will help you understand this, help me at least. Marla Spivak, with her VSH work, has selected for colonies that have such a high expression of VSH that they jettison all of their brood. Not just the sick brood, not just the mite brood. Let's just get rid of it. I can't stop. I got to pull it all out. Too much of a good thing, right? Greg Hunt has not done this personally, but just before he retired, he was working with some genetics colleagues in other fields, and the gene for grooming behavior, which is what Greg does, getting mites, picking mites off yourself, is the exact same gene as found in mice. It's not a homolog, it's not comparable to, it's the same identical gene is found in mice. And these workers, these teams working on these mice, have had mice that groom themselves so much that they die. They die of exhaustion. They die grooming themselves out of exhaustion. Too much of a good thing. So this, which is called the rare allele model, in my opinion, gives the best evidence for why polyandry evolves. It's more than just genetic variation. Let's go out and get that little extra. Now I want to finish in the time remaining to me about how polyandry can be applied to beekeeping, and I think it's very tangible, very straightforward. And this is what I did on my 2-year sabbatical in York in 2012 and 13 using instrumental insemination. We created 2 classes of queens. One was inseminated with drones, 15 drones. Another group was inseminated with 30 drones, and another group was inseminated with 60 drones. No selection, We weren't selecting for anything. It was just drones. And then we exposed these bees to, we measured them for a while, and we found significant increase in brood production as polyandry increased. The more polyandry, the better it brood, but this is the real interesting one. Significantly fewer mites when polyandry increased from 15 to 30 or 60. Those are mite numbers, so lower numbers is good. The 30 and there was no selection. No selection. This is just simply casting a wide net. I think this is important for us in beekeeping, and this is kind of where my funding has been for the last, last 2 years. We have been doing a project with Debbie Delaney at Delaware. It's true. Keith Delaplane working with Debbie Delaney in Delaware. My lab crew has fun with that. And we have designed, this is our design. We have created 2 classes of polyandry and 2 types of drones. One pool of drones has been pre-selected for Varroa sensitive hygiene. The other group is wild type. They've not been selected for anything. We're interested in the interaction between the 2. Lots of groups have been out there selecting for VSH or grooming, and what if we combined that with the benefits of polyandry? So 9 and 54 drones of either VSH stock or wild type stock. Why 9 and 54? Because only 9 of our drone colonies of each type survived over winter, and we wanted them to be in multiples. Multiples. So let me go ahead and show you the results for these cells here in the middle, because that's where I think most of the interest lies. How can we get interaction? And the results, I'm going to go ahead and tell you, are overall promising, but a little unintuitive in some respects. When it comes to bee population, there's an interaction between VSH and wild type. Wild types, the brood population gets better, significantly better, when you have 54 drones. But the reverse trend is true with the VSH stock. I think this might be kind of an example of what I was talking about earlier. You lose things. When you're selecting for something, other things get lost. And you know, this seems to be the case with the VSH crew. Selecting for lots and lots of VSH and lots of drones carrying VSH, we lose drone population. Or worker population, I mean. And the trend is same with brood. Brood always parallels bee population too, by the way. But let's take a look at mites. And this is a significant result that's favorable. We get a synergistic interaction that's positive when you have VSH drones inseminated with 54 of them. This is a positive synergy. And I find this exciting because imagine a market in drone semen. We could all be trafficking in straws of drone semen. People who are selecting for VSH. Well, let's get drones that are pre-selected for grooming behavior or Russians. You know, we could exaggerate that with polyandry based on results like these. I do want to say, though, these data are just from Georgia. We're just now this year setting up the replicate in Delaware. So we're going to be, what you're seeing here are just half of the data set. Let me go ahead and show a couple other things. Brood survival, strangely, was highest in the VSH 54 group. Survival. Curiously so. And these are so new that I really haven't had time to chew on that one yet. We also looked at pollen diversity, and we would expect diversity to be higher with polyandry, and that does appear to be the case in both classes. Let me summarize all this for you in a different way that I think will help put it all together. Looking at this design, we were looking at different things, and in bee population we found the maximum populations in the VSH groups that were inseminated with only 9, and in the wild types those were with 54. So the behavior behaves differently between wild type and VSH. When we looked at brood, the parallel happened because brood and population are always parallel to one another, mirror one another. But when it comes to mites, we found a positive beneficial synergy for the VSH stock that was inseminated with 54 drones. For brood survival, we found an increase in those groups that were polyandrous. And in pollen diversity, we found an increase in those groups that were polyandrous. So we're kind of seeing a trend here toward more positive checks regardless of stock in the polyandrous 54 column, which is encouraging to the line of work what we're doing. But the one that excites me the most is this one about mites. this positive synergy for something that's really current and of importance to us right now, and that is Varroa resistance. So what I mean, the point that I mean to get across with polyandry is that it's not in competition with classical selection. I see it as an enhancement to classical selection. And I think it really argues for the importance of bringing artificial insemination more into mainstream in bee breeding. We not only want bees that are selected for VSH and grooming, we want, like one of the beekeepers in London once said, just want to fill her up with sperm from the good guys. And he also asked, when I was talking about the numbers that we were using, like 60 drones, and he said, but won't she pop? But we do, it's a good question, but we collect all the drones, we mix them all up in a capillary tube, and then we draw from that pool and inseminate with a standard volume for each one. So this, I think, is useful, and it's very closely aligned with the evolutionary biology of the bee. And with that, I think I'm done talking, so thank you for your attention.